Hello students, in today's lab we are going to learn about plasmid DNA isolation by alkaline lysis method. So the aim is to isolate plasmid DNA by alkaline lysis method. In this experiment we make use of three solutions. Solution 1 that contains glucose, EDTA and tris. Solution 2 that contains 0.2 normal NaOH and 1% SDS. And solution 3 that is 3 molar potassium acetate solution containing 11.5 ml glacial acetic acid. We also need isopropanol for precipitation of DNA and LB broth for starting our uh, E. coli culture containing a suitable plasmid. So coming to the principle of the experiment, in the first step we centrifuge uh, the cells in media in order to obtain a cell pellet. We then resuspend the cell pellet in solution 1 that contains glucose, tris, and EDT. Glucose here functions to maintain the osmolarity. Tris acts as a buffer, and EDTA chelates divalent metal ions. So these metal ions are required by nucleases for their activity. So EDTA basically acts as an inhibitor of nuclease, protecting the DNA from degradation by activity of nuclease. Solution 2 contains NaOH and SDS. SDS is basically an anionic detergent and will denature the proteins by imparting overall negative charge to the proteins. This results in cell lysis. NaOH over here functions to create alkaline conditions. So, after cell lysis, in presence of alkaline conditions, DNA will denature. Both genomic and plasmid DNA will denature here. Genomic DNA is a linear double-stranded structure. On denaturation, it gets converted into single-stranded DNA. Plasmid DNA is supercoiled in nature, and hence, after denaturation, also, although the two strands they will uh, separate out from each other. However, because of the supercoiled nature of the plasmid DNA, the two strands of the DNA they still stay entangled with each other. On addition of solution 3, that is potassium acetate solution, the acidic conditions are restored. That leads to again conversion of double-stranded the single-stranded denatured DNA to double-stranded. So during renaturation of the genomic DNA, since it is large and linear structure, a part of it renatures with its own double-stranded DNA, and part of a DNA renatures with other single-stranded DNAs that results in formation of a larger aggregate. While plasmid DNA, since it is in supercoiled form and is small in size, so it is able to quickly renature on addition of solution 3, and you get intact double-stranded plasmid DNA that is formed again. As compared to the genomic DNA that forms large aggregates because of um, renaturation with different or reannealing with different strands, resulting in formation of larger aggregates. After this stage, when we centrifuge our lysate, genomic DNA that forms large aggregates will come in the precipitate along with other debris. And in the final step, we make use of isopropanol for condensation and precipitation of DNA from the supernatant. Coming to the protocol, so first we need and culture of E. coli with suitable plasmid in presence of a suitable antibiotic such as ampicillin can be used. 1 ml of the culture is then aliquoted into append of tubes and centrifuged 10,000 rpm for 5 minutes. This will result in separation of the supernatant, and the pellet is formed at the base of the tube. The supernatant is discarded and the pellet is resuspended in 100 microliters of solution 1. We can pipe it here or also vortex in order to bring full resuspension of the bacteria in solution 1. Then we will add 200 microliters of solution 2, that is NaOH SDS solution, and mix by gentle inversion. After that, so here cell lysis happens, and your it can be observed by formation of sticky thread-like strands when we open the Eppendorf tubes. Followed by this, we will add 150 microliters of solution 3, that is potassium acetate solution, to the lysate and mix by gentle inversion. So at this stage, the genomic DNA forms the aggregates, larger aggregate, and the plasmid DNA is able to denature at this stage. Then we centrifuge at 10,000 RPM for 10 minutes at 4 degrees Celsius. We recover the supernatant, and the precipitate over here basically contains all the aggregated plasmid and other cellular debris, which come in the form of a white precipitate. So we recover the supernatant in a fresh tube, and we add equal volume or twice the volume of isopropanol. We mix it thoroughly by inversion and incubate at 4 degrees Celsius for 1 hour. This is followed by centrifugation at 10,000 RPM for 10 minutes at 4 degrees Celsius. Here, isopropanol is going to help precipitate the plasmid DNA. We then remove the supernatant, and the pellet is going to contain the plasmid DNA. We rinse the pellet in 70% ethanol and air dry it for about 10 minutes so as to allow the ethanol to evaporate. We then resuspend the pellet in PE buffer or sterile distilled water, about 10 microliters of which can be used. We can also treat with enzyme RNase to remove RNA contamination, and then we'll analyze the gel by agarose gel electrophoresis. Coming to the observations after agarose gel electrophoresis, we can see that the 1 kb DNA ladder has separated and it represents bands of different sizes. This is the band of plasmid DNA, which runs around 2.5 kilobase pair. We can also see some bands of genomic DNA contamination here. In some wells, we can see some trailing, which is because of the degradation of the genomic DNA, where you can see these bands at different positions. As a result, you can see a trailing kind of image. You can also see the large blob that is present towards the base, that is RNA contamination. On treatment with enzyme RNA, this is not visible. RNase treatment is basically required if we are using the plasmid DNA for cloning purposes. So in our results and conclusion, we can see that we were able to isolate plasmid DNA, which ran around 2.5 kilobase pairs. We also observed genomic DNA contamination, and RNA was also. So that's it about plasmid DNA isolation. Kindly like, share, and subscribe so as to get direct update on uploaded videos. Thank you.